Okay, there I got to pretend to be Ed, and that was challenging, and I guess I survived, and I guess you survived. So now I get to pretend to be Kelly, okay? So at our last talk, she did her first run of this, at our last, GBS workshop held in Cornell, Kelly added this new talk about imputation strategies. So she, as a grad student in Ed's lab, she's been working on imputation. So she developed an algorithm that she calls fill-in, fast inbred line library imputation, and probably should have Peter Bradbury's name on here as well, but I don't, because it was her talk. And but he developed this thing he's calling full sieve family haplotype reconstruction, or I guess full hap, full sieve family haplotype. That's what he calls it. So I'm going to talk about those things. So first, in general, imputation. So imputation is the projection of genotypes onto missing data, filling in the ends. So, and now that we're doing genotyping by sequencing, we're producing lots of data with low coverage, especially when we use APK1 as an enzyme. So there's a need for imputation that can handle data sets with lots of missing data. But doing it low coverage is cheaper. So that's why we want to be able to cover lots of individuals at lots of markers. So the strategy in the Buckler lab was, we want to do GWAS. We want to be an LD with causative polymorphism. So we need to do a lot of markers. And we need to do them in a lot of individuals. So we have to go, we don't really have the choice. We have to go at low read depth and then use imputation to fill in what we're missing there. So if you have all this missing data, it's a problem for association mapping and genome predictions. Missing data can reduce the power and can reduce the accuracy, can give you false positive spurious results. So you could be either filling in the missing data on an existing data set with a certain set of markers and you're using those same set of markers, or another situation is projecting higher density data onto lower density data. Okay, and that can happen. Let's see, hang on a sec. Right, sorry, I'm giving someone else's talk, so I'm getting a little, right. Projecting higher density to lower density data, that can happen like in humans, they have the 1,000 Genomes project, where they've done whole genome sequencing on 1,000 individuals. So they're and they try to pick those individuals to be representative of human variation. So that gives them 1,000, and then they phase those things and they end up with 2,000 haplotypes that they can then project onto everything else. So that they're doing the very high density markers on those 1,000 genomes, essentially painting those genomes. And then they do lower density markers like a human SNP chip on hundreds of thousands of individuals, and then they project those higher density sequences onto those other ones. So that's a form of imputation. We're also doing that in maize because we have maize HapMap1 that Rob mentioned this morning and HapMap2 now. So, and we're moving towards HapMap3, which will be 850 maize lines all done with whole genome sequencing at high density. So we could project that. So our strategy is to have those 850 lines where we painted chromosomes and then have our GBS done in up to 90,000 maize is what we're heading towards. And then we can project those painted chromosomes, those painted haplotypes onto those 90,000 lines. So that's where we're going. So there's those kind of two ways of imputation. So generally when you're imputing, you have to generate haplotypes to start with. And so you can use IBD segments and clustering to help identify those. Or like I mentioned, the 1,000 genomes is, if you're working in humans, you've already got those haplotypes ready from the 1,000 genomes work. Right, so imputing back onto the target samples where the missing data is usually involves some kind of HMM model. So the current algorithms that are out there for imputing, most of them were designed for humans where there's low structural variation, and most of them were designed for obligate outcrossers where there's Hardy-Weinberg and you expect those Hardy-Weinberg expectations are used in the calculations and things. And most of them are quite slow. They're not able to impute 40,000 maze lines that Kelly's been working with. She tries to she'll try an algorithm that's out there, and let's try this program, and a week later it's still running, so it's just not practical. But one she did find that worked really well, that's existing algorithm, doesn't come from our group, it's called Beagle version 4. So in particular, version 4 was a big leap forward for dealing with GBS type data sets where there's lots of markers and lots of missing data. So in this talk I'll present results of Kelly's algorithm and Peter's algorithm compared to Beagle 4. Okay, so basically in the Buckler lab, Kelly and Peter and others together have been working on new approaches just for crop plants. So Peter's is called the full Civ family haplotype, and it's trying to finally map these recombination breakpoints. So it's really an HMM algorithm, and I actually talked about it yesterday as well. So he developed that for application in the maize NAM families. And we're doing that because we want to fine map QTL, and we want to understand patterns of recombination across the genome. And Kelly's is called fill-in, which is a generalized approach for all populations, not just full Civ families or biparental families, but even for unrelated things. And this is optimized for GBS type data, very large amount of markers with a lot of missing data. And it leverages inbred lines, which we have a lot of our maize lines that we're genotyping are inbred, some of them aren't. And but it also finds IBD, identical by descent, segments that could even be in unrelated individuals, and uses those as well to find haplotypes. So I actually talked about this a bit. So the HMM is helpful to find transitions between the different haplotypes or where the recombinations are. And I'm not going to go too much through this. I'll skip over it. I showed that yesterday. And I showed that yesterday. So mostly you're trying to pick out where are these recombination breakpoints between the different haplotypes, and also call heterozygotes, and also correct errors, right? So that's going to deal with map expansion.
I just want to make sure that the group, I think this is a really useful diagram to see the kind of data that you'd like get a gut feel for the swapping back and forth in a segment that would kind of indicate ahead. Do you guys have that gut feeling? Do you understand how you can look at this data and kind of interpret it that way? Yeah? I think this is a really kind of a key point, because when you're starting, if you don't have this imputation methods, you want to look at your data in such a way that you can kind of say, oh, maybe you can go and do some of it by hand, as you did early on, right?
Right, yeah.
So it's nice to have these algorithms, but it's really better to have that gut understanding of, geez, when I look at GBS data and it looks like this is what it likely means.
So this was initially developed for a biparental context, but imagine if you had those 1,000 genome haplotypes out there and you were trying to say, you knew, then you could pick, it looks like these ones are the two parents, but where's the recombination breakpoint between them? That's what this is used for in that context. So here's like Kelly's schematic of fill-in. So it has these various steps. So the first step is to generate blocks of haplotypes on some small chromosomal chunk. I think she works with 8,000 sites, 8,000 SNP chunks of the chromosome. So she looks across all the 42,000 taxa in a window of 8,000 sites long, and she looks for duplications of that haplotype across these things. So where it's shown up in multiple individuals, and they don't have to have all the same data in common, but they have enough data in common that you can diagnose that must be the same haplotype. So here's one in one individual that's orange, here's another one that's orange, and when you find that multiple times, you can combine it all together in this haplotype for that little window, and do that across all the possible haplotypes. So you don't even necessarily need inbred lines, but inbred lines are really going to help this work better. You at least need some level of homozygosity out there to identify haplotypes. So once you have this set of haplotypes, then you look at each individual at a time and try to find, for that window of the genome, and try to find the haplotype that's closest to that. So if you find one that's really close across the whole window above some threshold, then you just impute and say, oh, that's what my individual is, and fill in the entire haplotype. Or maybe it's a combination, in that window, it's a combination of two parents with a breakpoint in between. Well, that looks just like that Viterbi stuff, the HMM. So you could try to figure out with the HMM where that breakpoint is. So this would work if you're searching through where's the best haplotype, and you find two that each of them, they look pretty good on their own, but neither of them is really great. Neither of them really explain the full thing, but there's two of them that sort of beat out all the others as the closest to the thing you're trying to impute. So then you take those as the two putative parents and try to figure out use the HMM to try to figure out where the cut point is. So if you're successful doing either of these steps, you're done. But if you're not, then you move on, and then Kelly breaks that chunk into smaller pieces and basically repeats this process. Either finds one haplotype that explains the whole chunk, or finds a combination of haplotypes that explain it, and uses the Viterbi to say where is the recombination. If that doesn't work, then start to look for heterozygous combinations. So two parents where together they'll explain that chunk and call it a heterozygous for the two haplotypes. So this is where you can start to deal with heterozygous individuals like maize landraces that are not inbred lines. And if that works, great. If not, then she doesn't impute anything. If she can't find something that explains it well, then she leaves it missing, which is good if it really is biologically missing. So that's the basic explanation of fill-in. Any questions there? Yes?
Sorry, in terms of what's actually being imputed here, is it missing data from genotype loci or missing data from non-genotype loci or both?
In this case, it's missing data from genotype loci because it's just within GBS. But it could be applied in either way, I think. Yeah. But you'd have to have at least some data in common. So you could have the GBS data and the HapMap data, and trying to project the HapMap data down onto that GBS. So they'd only have the GBS would be a subset of the data where the haplotypes are filled in, but you could still project it down. So that's where we're going with this, to be able to do GWAS in 90,000 maize plants, basically. So, any more questions? So when you're doing imputation, you can figure out whether you're doing a good job or not by masking the known genotypes. But since we have low depth, we don't really know when they're heterozygotes. So what Kelly did was just take those individual genotypes where the depth was 7, exactly 7, which is pretty good depth. So, and she used those as known genotypes. So most of the time, it's going to, when you have a depth of 7, you're going to correctly call a homozygote or a heterozygote. It's not often you're going to flip 7 heads or 7 tails in a row. And so she did that. So she used those, and she masked those, that subset with 7 depth exactly, or at least a random sample of those. And then she did the imputation and then unmasked those and asked how often did I get it right. And this doesn't come out well here, but this basically shows that the distribution of read depth across the SNPs that have sites like that is the same as a random collection of SNPs from the data, so it's still representative. So she calculated accuracy by R² between the known and imputed genotypes, so the correlation. And she also ran Beagle 4 for comparison. I'm not sure what she means here, so I'll skip that. And this has been totally messed up shifting to this machine. I think the sizes are still there. So this is like comparing the FSF HapMap thing, which is for Peter's method for full SIB families, versus fill-in on full SIB families versus Beagle. And the main point is this one, the one that's designed for full SIB families does the best, pretty much. The triangles are represented how much of each genotype actually gets imputed and how much is left missing. So that's the axis on the right. And so the full Peters algorithm, FSF HapMap leaves some missing. Fill-in leaves a lot more missing, where Beagle always imputes over everything. So when a thing is truly biologically missing, you don't always want to impute over it. So that's one drawback of the Beagle. Then Kelly compared 3 different types of data sets of unrelated things, either temperate inbreds with low diversity and low heterozygosity, or a diversity panel of inbreds with higher diversity, but they're inbred still, or diverse land races. So I'm just going to show her comparisons of these 2 here. And yeah, that's not bad. So she used just a really naive imputation method where you just put in the major allele, or you put in the allele frequency. What she would do is draw at random based on the allele frequency which genotype to call it. That's what that is. And on these graphs, you're doing a good job when everything is on the diagonal line. The more stuff you have out here, the more you're getting wrong. So there's known genotype and predicted genotype. So this naive one makes mistakes quite a lot, like this is wrong. Whereas Beagle doesn't make that many mistakes and calls a lot of heterozygotes. Maybe overcalls them, because there's these, well, whereas fill-in does a great job on the homozygotes, but not such a good job on the heterozygotes. But the R-squared for fill-in is really good. So this is inbred panel that's diverse. And also fill-in leaves some data when it's not, doesn't call the heterozygotes when it's not sure, which might be a good thing in some cases. With diverse land races, Beagle does a lot better. Land races are highly heterozygous, so Beagle does better than fill-in. Is the main message here. This one I think is comparing fill-in to Beagle and showing that it does a better job whenever we have minor allele heterozygotes. So the orange and the red, it does better than Beagle for minor allele homozygotes, I'm sorry. But it doesn't do as good a job as Beagle on heterozygotes. So the point of this slide, there's a bunch of numbers here, but the most important thing is this thing. When Beagle and fill-in both agree, then you get improved accuracy altogether. So if you wanted to have the most accurate you could use, you run both and then pick the calls where they both agree. And yeah, all these slides are getting messed up. But this still shows the trend, so it's fine. So the one thing about fill-in is it's much, much faster than Beagle 4. It's kind of optimized for speed. So even imputing lots of taxa, 5,000 taxa, it doesn't go up as fast the computation time compared to fill-in doesn't take as much time as Beagle. So should you use it, it's going to be implemented in TASL 5 with documentation and whatnot. They're submitting a paper on it. It's good for very large inbred low coverage data sets like GBS and crop plants. If there's a lot of biologically missing data, then either fill-in or full subfamily HapMap is going to be less likely to over-impute over top of those really missing chunks of DNA. If you're working with full subfamilies, definitely use full sub HapMap. If you're confident in the parental genotypes, which means you believe the crosses are good. But if you're working with really heterozygous stuff, probably Beagle 4 is still better. And these are the folks that did most of this work. Especially, we should, as I kept saying, Peter's name, he was very important in this. And that's all I have for this. Any questions?
Everyone's stunned.
Pardon me?
Everyone's just stunned.
Oh, yeah, yeah. That's okay. This is the end of the week for me, so it's been a long week. I'm stunned too.
Short questions. So before you do imputation, do you put a threshold? If a large percent of missing value, just disregard, or doesn't?
So the question is, do you put a threshold on whether or not to impute based on the amount of missing data at a particular locus?
I think that threshold would have been the filters that went into producing the genotypes in the first place. So I don't think there's any additional one for that. But it would kind of make sense to maybe look across a segment, you know, versus look across a bunch of contiguous SNPs in a segment and compare it for that individual, whether that coverage was really low across multiple SNPs in a segment compared to the entire genome, but just for that one individual. And if it looked low, maybe don't impute because maybe there's missing stuff in there.
It's good idea to discard imputation itself can cause conflicts.
Yeah, that's what the over-imputation is problematic, and that's one of the messages Jeff was, yeah.
A question about tassel really, rather than this particular topic. Can tassel be used in these kinds of modules and elements of tassel be used on GBS data generated through other means?
So let me try to rephrase your question. Are you talking more about the post-SNP calling portions of tassel?
Really, it's well, even the SNP calling component, I mean, if we just get sequence data.
Well, so the SNP calling is, of course, that Jeff has been working on is really all about GBS data. So it's not built for a whole genome sequence or anything like that. Such as exome capture. It doesn't have an enzyme cut site, right? So that's really not gonna work for the SNP caller that Jeff's built, right? But other components, any HapMap data set with SNPs that you can just pull into Tassel, and you'll see this a little bit later today, can use the tools that are there, right?
Yes.
I mean, it doesn't matter whether they were generated, those HapMap files were generated by Tassel or not. It's a standard format, so you can pull it in and manipulate.
I think they're going to keep the Plink format going forward, so you can bring in Plink format data or VCF as well. We're moving towards more and more support of VCF because it's a community standard and also holds a lot of annotations in it. And of course, we're developing our own HDF5 format that can be used.